Hello, welcome to this presentation on the regulation of respiration. Now, why is it important that the respiratory system is tightly and accurately regulated? The regulation of respiration, by and large, the main objective is to ensure that the partial pressure of oxygen, the partial pressure of carbon dioxide, and the pH of blood. In particular, these parameters measured in arterial blood should be kept within normal physiological ranges. Why? Because we want to ensure that there is normal functionality of the body. Now, these tend to get deranged, especially if the physiological state of the body has changed. In the case of exercise or in diseased states, you find that these factors get deranged and therefore they must be brought back to their normal physiological ranges. How do we do that? There are certain mechanisms that kick in physiologically to maintain these within normal physiological ranges, and that is what we are discussing today. So we just want to see how the body is able to bring back the partial pressure of oxygen, the partial pressure of carbon dioxide, and pH back to normal when there's a derangement in the body. Now, the major player in this regulation is the nervous system, and it is the final player to execute the change that is desired, whether it is to bring up when these parameters go down or when it is to bring down when these parameters go up. What plays a role by and large is the nervous system. Of course, the nervous system plays this role in two ways. There's voluntary control and involuntary control. Voluntary control involves the cerebral cortex, whereas involuntary control involves the pontine and the medullary respiratory centers. So these together are able to regulate respiration. Now, this here is just a schema summarizing the whole process of regulation. So, as earlier mentioned, the main objective is to maintain the normal levels of what? The partial pressure of oxygen, the partial pressure of carbon dioxide, as well as the pH of blood in where arterial blood. It is arterial blood where we are measuring this. Now, this regulation has got three basic elements. We have there are sensors, then we have the central controller in the brain, and then we have the effectors. These sensors include the chemoreceptors, the pulmonary receptors, as well as other receptors. The chemoreceptors are of two types. We have the central chemoreceptors and the peripheral chemoreceptors. The central chemoreceptors are found in the brain. The peripheral chemoreceptors are found in the outside the brain, and to a larger extent, the peripheral chemoreceptors that we're interested in are the aortic bodies and the carotid bodies. Of course, there are other peripheral chemoreceptors, but these are the main peripheral chemoreceptors that we are talking about. We also have the pulmonary receptors. These include receptors such as the stretch receptors in the lungs, as well as the juxtacapillary receptors in the lung parenchyma. Then these sensors, once they detect these changes, be it a low pH, a low partial pressure of oxygen, or a high partial pressure of carbon dioxide, once they detect those changes, they are going to send signals to the brain, the central controller. Now, in the brain, which centers are important in playing this function? We have the brainstem. So in the brainstem, there are two centers, the pons and the medulla. Then the cortex. So the cortex is involved in voluntary control. Voluntary control meaning doing things like breath holding. Like when you decide to voluntarily hold your breath, it means what is enabling you to do that are fibers coming from the respiratory centers in the cortex, because the cortex is for voluntary functioning. Then the brainstem, these ones play the biggest role in from the brain to regulate respiration. So there are centers in the pons and centers in the medulla that play a role in the regulation of respiration. Then once these interpret the signals that have been sent to them, they send back input via certain fibers, nerves. By and large, it is via the phrenic nerve as well as the intercostal nerves to these effector muscles. So these effector muscles include the diaphragm, the abdominal muscles, and other muscles of accessory muscles of respiration, which may be the sternocleidomastoid muscle as well as the scalene muscles. So these, they play a role in effecting the change that is desired. So this is just a schema summarizing the three basic elements of what? Regulation of respiration. Now, like it was earlier mentioned, the neuronal control of respiration involves both voluntary and involuntary control, and involuntary control is under subconscious control. Then the diaphragm muscle as well as the intercostal muscles are the primary muscles of what? Respiration. And they are stimulated by groups of neurons found where? In the pons and the medulla. So the two main respiratory centers in the brain include the two main respiratory centers in the brain for involuntary control include the pontine respiratory center as well as the medullary respiratory center. The pontine respiratory centers, these ones controls the rate and pattern of breathing. The pontine respiratory centers controls the rate and pattern of breathing. That is the pontine respiratory centers. Then we also have the the pontine centers are made up of two areas, the apneustic and the pneumotaxic center. So the apneustic center causes what? Inspiration. The pneumotaxic center just controls the fine tuning of the respiratory rate and the respiratory depth. In other words, it controls transitioning from what? Inspiration to expiration, because it sends inhibitory signals to the apneustic center to inhibit inspiration or to stop inspiration and therefore allow expiration to do what? To take place. That is what the pneumotaxic center does. So it controls transitioning from inspiration to expiration by sending inhibitory signals to the inspiratory centers. Yeah, that is the pneumotaxic center. It sends inhibitory signals to the apneustic center to inhibit inspiration. And we also have the medullary. Centers, respiratory centers. They control the basic rhythm of what? Breathing. That is what the medullary centers do. So they are made up of two groups of cells, the DRG, which is the dorsal respiratory group of cells, and the VRG, which is the ventral respiratory group of what? Cells. Now, if you look at this diagram just on the side, this one shows the two centers. That is the brainstem. So this is part of the brainstem, and in the brainstem, this is the pontine. So the whole section here that is being shown is part of the segment of the brainstem. Then we have got two centers. So this bulging center here, that looks like it's bulging, or this outpouching here, these are the pons. Then where these two, the blue and the green one, are, this one is the medulla. This is what the medulla is. Now, if you go back to the pons, you can see that we have the pneumotaxic center there and the apneustic center there. Like we said, the pneumotaxic center sends inhibitory signals to the apneustic center, and the apneustic center is for what? Inspiration. But when it is inhibited, meaning it is going to stop inspiration and allow expiration to occur. Now, that is the apneustic center. It also causes what we call apneusis, deep inspiration. That is apneusis, or that is also a function of the apneustic center. Then when you move to the medullary center there, you have two groups. You have the expiratory center, which is the VRG, and the inspiratory center, which is the DRG. So the DRG, dorsal respiratory group of cells, causes inspiration. The VRG, the ventral respiratory group of cells, causes to a larger extent expiration and some degree of inspiration. So now how these two groups, respiratory centers work is that you have the apneustic center there, which when stimulated sends excitatory signals to the DRG, the dorsal respiratory group. Once the DRG is stimulated, it sends its output fibers, its output signals via these fibers. That is the phrenic nerve. You can see that it's going to the diaphragm there, and the intercostal nerves to the, in particular, the external intercostal muscles. So once there's activation or stimulation of the apneustic center, it also activates or stimulates the DRG, then which sends signals via these fibers, the phrenic coming to the diaphragm and the external intercostal muscles. So what happens is when these excitatory signals are sent here, they are going to cause what? They'll send action potentials that are going to stimulate muscle contraction of these muscles, the diaphragm muscle and the external intercostal muscles. Contraction of these muscles causes what? Ventilation. So it causes these to hyperventilate. Then on the other hand, we also have what? The pneumotaxic center. So this pneumotaxic center, when stimulated, it can send inhibitory signals to both the apneustic center as well as the DRG to inhibit inspiration. So stimulation of the apneustic center and the DRG causes inspiration, but then to inhibit the inspiration, when the pneumotaxic center is stimulated, it can send inhibitory signals to both the apneustic center as well as the DRG. Then we also have the VRG. So the VRG, the ventral respiratory group of cells, these ones send inhibitory fibers or signals to the internal intercostal muscles as well as other muscles of other accessory muscles of respiration, such as the sternocleidomastoid muscles, the scalene muscles, the abdominal muscles. Fibers are going to come from here to stimulate contraction of those muscles, and those muscles play a role in what? Forceful breathing. When there's forceful breathing where you need to expire forcefully, that's when these muscles are recruited to assist in the forceful expiration. So this is how these centers are communicating. Now, the dorsal respiratory group of cells, remember this one is for what? Inspiration, and it is found in the medulla. It contains the nucleus of tractus solitarius. So it contains the nucleus of tractus solitarius, which is responsible for the basic inspiratory rhythm. That's what this does. So it receives all input or impulses coming from the different chemoreceptors. The chemoreceptors, the mechanoreceptors, as well as the stretch receptors. All these have the capacity to send signals based on the information that they detect at their receptor site, then send their fiber information or action potentials through the various nerve fibers that come to feed the dorsal respiratory group of cells and cause the necessary action from it. Then we have the ventral respiratory group of cells. It also contains This one contains both the inspiratory and expiratory neurons. So it is made up of number one, the Botzinger complex, which controls expiration. Then we also have the pre-Botzinger complex, which contains the pacemaker neurons. So in other words and in other texts, you'll find that they say the pre-Botzinger complex is the one that functions as the pacemaker of the respiratory system. It sets the respiratory rate. That is the pre-Botzinger complex. Then, apart from those two, there's also what we call the nucleus retroambiguous. This one controls both expiration and inspiration. Then we also have the nucleus paraambiguous, which controls inspiration and expiration. How? By controlling the laryngeal and the pharyngeal muscles. That is how the nucleus paraambiguous is able to control both inspiration and expiration, by controlling muscles of the upper airway. That is the laryngeal and the pharyngeal muscles. So this is what makes up the ventral respiratory group of cells. Yeah, so this is just a summary again of what has already been discussed. You have the medulla here with the pons and the pons and the medulla making up the brainstem. Like we said, of the acoustic center, you can see that it is sending excitatory signals to the dorsal respiratory group, which sends excitatory signals to the diaphragm and the intercostal external intercostal muscles to cause what? Contraction, that is inspiration. And if it is acting at a faster rate, you induce ventilation, hyperventilation. Then at the same time, you can see that there are inhibitory signals from the pneumotaxic center going to the DRG. At the same time, there's communication between the VRG and the DRG. Some communication can be excitatory, some other times it can be inhibitory. But from the VRG can also receive input from the pneumotaxic center to induce what? Expiration. As you can see, there are fibers that are coming from the VRG going to the accessory muscles of respiration, as well as the internal intercostal muscles. So these are fibers coming from the VRG that cause the expiration. Now, we look at the chemoreceptors now. So there are two chemoreceptors, two classes of chemoreceptors that we are going to look at. There are central chemoreceptors and peripheral chemoreceptors. These receptors tend to respond to alterations in what? The partial pressure of oxygen, the partial pressure of carbon dioxide, as well as the pH. Changes in these are picked up by specific receptors that we call the chemoreceptors. And these may include the central chemoreceptors, the peripheral chemoreceptors, or the stretch receptors, irritant receptors, juxtacapillary receptors, the proprioceptors, to mention but a few. Now, the chemoreceptors, there are two main types. We have the central chemoreceptors and the peripheral chemoreceptors. Now, the central chemoreceptors are found in the brain. Yeah, so they are found in the brain. So these ones, the major stimulant is what? A buildup of protons in brain tissue. So a low pH in brain tissue activates the central chemoreceptors. They are found in the brain. So the major stimulant of central chemoreceptors are found in the brain. The central chemoreceptors are located in the brain, and the major stimulant is a low pH in brain tissue. Now, how does this pH build up? So, if there's an increase in the partial pressure of carbon dioxide in arterial blood, what that will do is it is going to diffuse across the blood-brain barrier, and once it diffuses across the blood-brain barrier, there's going to be a reaction between carbon dioxide and water to form carbonic acid, which is going to dissociate to produce what? Bicarbonate and protons. Then this is happening in the brain tissue. So remember, there's a buildup of carbon dioxide in blood, arterial blood. So once the carbon dioxide builds up to abnormal levels, it starts diffusing. Because it is carbon dioxide is lipid soluble, it will diffuse across the blood-brain barrier into the brain tissue. Once it enters the brain tissue, this reaction occurs. It will react with water to give us carbonic acid, which is going to dissociate to give us protons and the bicarbonate. The protons that begin to accumulate and build up stimulate the chemoreceptors. Once these central chemoreceptors are stimulated, they send signals to the DRG. And remember that the DRG, the dorsal respiratory group, does what? It causes inspiration. So if it is going to be stimulated, and it is going to be stimulated at a faster rate, it is going to cause an individual to hyperventilate. So if somebody begins to hyperventilate, it means they're going to start breathing in and out faster. What does that do? It starts removing the excess carbon dioxide that built up. So if you start removing the excess carbon dioxide at a faster rate, it means the amount of carbon dioxide starts coming down. If the amount of carbon dioxide starts coming down, it means this reaction has to begin to do what? To reverse, going back. So if this reaction begins to reverse, the stimulation of the central chemoreceptors begins to do what? To reduce also. So as the stimulation reduces, it means the activation of the DRG also goes down, meaning the person no longer or reduces the rate of ventilation, and it starts coming down until normal levels are brought down and back to within the physiological ranges. So these are the central chemoreceptors. Again, the major stimulant of central chemoreceptors is brain tissue pH. So pH is the major stimulant for what? The central chemoreceptors in what? Brain tissue. So brain tissue pH, low pH in the brain tissue is what stimulates these chemoreceptors. Not a low pH in blood, no, but in brain tissue. That is what is going to stimulate the central chemoreceptors. Yeah, so it means that if there's reduced partial pressure of carbon dioxide in blood, there's reduced levels, no stimulation of the central chemoreceptors, then no signals are sent to effector muscles, hence decreases respiratory rate. This now begins to allow carbon dioxide to begin to accumulate. If it had reduced, it begins to accumulate and going back to normal levels. Now, we move to the peripheral chemoreceptors. So the two peripheral chemoreceptors involved in the regulation of respiration are what? The carotid bodies that are found at the bifurcation point of the common carotid, and the aortic bodies along the aortic arc. So these ones, they respond to changes in what? Carbon dioxide, oxygen, and proton levels, with the major stimulant being what? Hypoxia. The major stimulant being hypoxia. That is a partial pressure of oxygen less than 60 millimeters of mercury. So these are the two major stimulants, the aortic bodies and the carotid bodies, with the major stimulant being partial pressure of oxygen less than 60 millimeters of mercury. Now these cells, or these bodies, contain two types of cells, namely the glomerulus cell, which is a type 1, and the sustentacular cells, which is a type 2. Yeah, which is a type 2. The glomerulus cell is a type 1. The sustentacular cell is a type 2. The function of the sustentacular cell is just to provide structural support to the carotid and the aortic bodies, as well as ensheathing the type 1 cells. That's the main function of the sustentacular cells. Then the glomerulus cell. So the glomerulus cell, is here. The glomerulus cell contains, or is made up of, these potassium channels. these potassium channels. These channels are sensitive to low partial pressure of oxygen as well as a low pH. So once there's hypoxia or a low pH, these potassium channels, sensitive channels, are inhibited, so they close up. When they close up, there is going to be buildup of positive charge within the glomerulus cell, which is going to open up the calcium channels, the voltage gated calcium channels. So once the voltage gated calcium channels open up, there's going to be an influx of calcium into the glomerulus cell. So this influx of calcium is going to cause fusion of these vesicles that are in these vesicles. So these vesicles contain dopamine. So an influx of calcium into the glomerulus cell is going to cause fusion of these dopamine containing vesicles with a membrane. Once there's that fusion, there's going to be exocytosis, and once exocytosis occurs, these vesicles contain the dopamine, which is going to be released outside. Once dopamine is released outside, it is going to stimulate these nerves. So in the aortic bodies, this dopamine stimulates the nerve terminals of the vagus nerve, whereas in the carotid bodies, this dopamine stimulates the nerve terminals of the glossopharyngeal nerve. Both these nerves send their fibers toward the nucleus of tractus solitarius in the DRG. And remember what the DRG does? It causes inspiration. So meaning that if there was a low pH, what you want is to increase the rate of ventilation so that you can breathe in faster to increase the amount of oxygen that is present. So that is what is going to happen. So because you are stimulating these fibers, they're going to stimulate the DRG, the nucleus of tractus solitarius. The nucleus of tractus solitarius is going to send its signals down via the phrenic and the intercostal nerves to cause what? Contraction of the diaphragm muscle and the external intercostal muscles. Contraction of those muscles causes an individual to hyperventilate. So this is how these the aortic bodies and the carotid bodies are able to control low partial pressures and bring them low partial pressures of oxygen and bring them up. Yeah, then we also have the stretch receptors. So for these ones, the stretch receptors are responsible for what we call the Herring-Brewer reflex. So now what happens is, if you see here, we have the so this is the lung with a pulmonary stretch receptor. So what happens is, when you inspire, this lung has got an optimum capacity that it can, the amount of volume of air that it can accommodate. Beyond that capacity, any amount of air that begins to enter begins to stretch the lungs, and it is at that point where expiration has to kick in. Now, how does expiration kick in? It's because of these stretch receptors. So when you inspire enough volume of air, beyond that, you stretch the receptors. Once the receptors are stretched, the pulmonary stretch receptors are stretched, they are activated, and they send signals to the what? Nucleus of tractus solitarius. And remember what it does. So these fibers that are going to, or these information signals that are going to the nucleus of tractus solitarius, are inhibitory in nature. So they inhibit the nucleus of tractus solitarius. At the same time, they are going to send excitatory signals to the expiratory centers in the what? Dorsal respiratory group. I mean the VRG, the ventral respiratory group. Also, they inhibit the apneustic center, but they also stimulate the pneumotaxic center. Stretch receptors, once stimulated because of volume of air that is going now beyond the capacity of the lungs, the stretch receptors are activated and they send inhibitory signals to the DRG, excitatory signals to the VRG to cause expiration, as well as excitatory signals to the pneumotaxic center. Remember, the pneumotaxic center inhibits inspiration. They also inhibit the apneustic center, meaning will inhibit what? Inspiration. Because they're sending inhibitory signals to the DRG, they are inhibiting inspiration. What does that mean? You now transition from inspiration to expiration, hence expire the air. That is what the stretch receptors are doing. They are just ensuring that you do not overinflate the lungs. This whole phenomenon that happens is what we call the Herring-Brewer reflex. The Herring-Brewer reflex. Then there are also irritant receptors. These ones are triggered by irritants and they send the signals via the vagus nerve, but their response is not by increasing ventilation, but by causing what? A cough or a sneeze reflex. Then we also have the juxtacapillary receptors. These ones are of two types, of the G and the C receptor fibers. They're located in the lung parenchyma and the interstitium. What do they detect? They respond to fluid accumulation. For example, in pulmonary edema, pneumonia, and left ventricular heart failure. So these ones, they are able to detect the fluid there that builds up. That is why if you find patients that have got pneumonia in heart failure, you find that the respiratory rate is elevated. Why? It's because these signals, these receptors are activated or stimulated constantly because of the presence of fluid in the lung parenchyma. So they are going to send signals to the DRG and as well as the apneustic center to increase the ventilatory effort to try and bring in more oxygen because of the presence of fluid. There's impaired gaseous exchange. Because gaseous exchange is impaired, there's a buildup of carbon dioxide and also a decrease in partial pressure of oxygen. So in an effort to try and increase that, these receptors are responding by causing an increase in ventilation. Yeah, so that is. Then we also have the proprioceptors. So the proprioceptors, these ones are located in the tendons, the muscles, and the joints. So because they are located in these areas, it means that what stimulates these is tension that builds up in the tendons as well as movement. So when somebody is moving, these ones are activated and they send their signals via the dorsal root ganglion, and then they enter the posterior gray horn and then through the dorsal white column, which comes up via the dorsal spinal thalamic tract. Then they decussate to form the medial lemniscus, which sends signals to the DRG. That is the proprioceptors. So in other words, these are found in the joints, muscles, and tendons, and they detect movement. That is why if you notice that when you start performing activities that involve you to move, and if you're moving very fast, you also begin to ventilate very fast and respire very fast. Why? Because there are also these receptors that are sending stimulatory signals to the brain to say increase the rate of ventilation, because we do not want to create an oxygen debt due to an increased consumption of oxygen. So you start ventilating fast so that because you're increasing the depletion of oxygen, you are quickly also replacing it. That is what these receptors are doing. They are activated due to movement. And then there is also voluntary control, and voluntary control is under the influence of what? The cerebral cortex. So voluntary control comes from the cerebral cortex, and the fibers are mainly for breath holding, but these sends their fibers and signals directly to the ventral gray horn. They bypass the medullary respiratory centers and they directly stimulate the respiratory muscles, that is the diaphragm and the external intercostal muscles. They do not pass through the brainstem centers. They directly go to the spinal cord and the ventral gray horn and send their fibers out to their signals to the primary respiratory centers. So as you can see, as you can try there, you can try to hold your breath and see, because you're able to do that because you've got fibers coming from the cerebral cortex. That is the voluntary control. Now, voluntary control, in the initial phases, states of it overpowers involuntary control. However, the longer you hold your breath, for example, when you decide to hold your breath, the longer you hold your breath, the more power the involuntary control begins to build up. And it is going to build up so much effort that it is going to overtake the involuntary control. So voluntary control is short-lived because involuntary control kicks in to overpower the voluntary control. Yeah. That is the end of our lecture. Cheers.